Welcome to our first video on giving a 10 to 15 minute scientific presentation. In this video, we'll explain how to create an introduction for such a presentation, one you might give in a laboratory discussion group or in a brief talk you might give to colleagues. Because you have only 10 to 15 minutes for your presentation in this format, you will need to be extremely concise, carefully choosing your words and your slides. These brief presentations are often more challenging than providing a longer presentation. This is the first of a two-part video sequence on a 10 to 15 minute scientific presentation. In this video, we'll focus on how to structure that introduction that sets the stage for your talk. In the second video, we'll focus on the body of the 10 to 15 minute scientific presentation: design methods, data results, and conclusions. The focus in that video is on the overall structure of the rest of your presentation. Note that the same patterns can also apply for a longer talk. There you expand the scope and or apply the patterns to a succession of research questions. The most effective introduction for an oral presentation follows a classic rhetorical pattern. Think of buckets where each rhetorical statement you make is placed in its appropriate bucket. In other words, you want the statements you make in your introduction to fit a particular pattern. And what are the appropriate rhetorical buckets and their sequence? There are four: context, complication, question, hypothesis. Use this CCQH pattern to shape your introduction. It guides the thinking of the viewer like one approaches a scientific problem, and it should take you less than a couple of minutes in your 10 to 15 minute talk. A good introduction for a 10 to 15 minute presentation puts the science problem in its appropriate buckets. That's CCQH pattern. The first C points to a context your audience understands and accepts. There's no dispute about it. It's a given. Then we get to the complications. These complications raise a compelling question. The complication is a state of uncertainty, of the unresolved, of the unknown, sometimes framed as a set of alternative explanations or an area where knowledge is incomplete or causes are poorly understood. And that complication raises a question which is implied or explicitly stated. And it's the compelling research science question that addresses that complication. And then finally we have the hypothesis. So your question offers a testable, clear hypothesis that when researched and analyzed will advance knowledge and understanding. And the introductory rhetorical pattern is like a funnel, moving from the big and the general to the narrow focus of the hypothesis. Your context gives the big picture, and it's really important to do this because with a good context, you connect with the audience. Your audience may or may not know the specific components of your research, the specific technical details. This way they see how it relates directly to them. Next, you have the complication. That's the issue, the problem, the hurdle, the difficulty. The complication tells us what's unknown or uncertain about the issues raised by that context. Then you have the question. The question is raised by that complication and should be answerable in a way that will resolve the complication. And then finally, we have the hypothesis. So the hypothesis focuses not only on the work itself, but the presentation. The presentation is designed to convey to the audience whether or not you've proven your hypothesis, disproven your hypothesis, or otherwise raised additional questions as a result of trying to test that hypothesis. Now let's look at an example based on how to structure an introduction. This example is based on a paper entitled "Amyloid Beta Protein Dimers Isolated Directly from Alzheimer's Brains Impair Synaptic Plasticity and Memory." That was published in Nature Medicine in 2008. You can also find a link to the paper on the Climb website. So here's an example of an introduction that follows the model using the paper we referenced on the previous slide. First, we have our context, the standard view. Alzheimer's disease is a rising threat to public health. It is distinguished from other dementias by abundant extraneuronal deposits of amyloid beta. Research shows that neuronal alterations can be induced in the brains of amyloid precursor protein transgenic mice by supraphysiological concentrations of synthetic amyloid beta peptides, by amyloid beta species secreted by cultured cells, or by mixtures of amyloid beta assembly forms. So this is the standard view. The linkage is commonly accepted. We can induce AD-type activity by placing synthetic amyloid beta into the brains of transgenic mice. Then we have our complication. Although these findings show that A-beta can alter synapse physiology in experimental models, the nature of pathogenic species from the human brain and demonstration of neurobiological effects haven't been shown. In other words, we know that synthetic and cultured A-beta has induced AD, but no one has used human A-beta and discovered its effects. Next, we have our question. Our experiment was designed to take soluble amyloid beta oligomers directly from the cerebral cortex of deceased subjects with Alzheimer's disease to induce Alzheimer phenotypes in normal adult rodents, measuring the effects on synaptic long-term depression and long-term potentiation, as well as dendritic density. So here we imply the question, could human A-beta induce AD in transgenic mice, and how would it do this? And then we have our hypothesis. We hypothesize that soluble A-beta isolated directly from human Alzheimer's diseased brains would potently impair synapse structure and function, specifically by the actions of A-beta oligomers and dimers. So we offer here a clear, testable hypothesis that when we research and analyze it will advance knowledge and understanding. So what follows is a single PowerPoint slide that organizes an introduction to the amyloid beta protein dimer problem in a different way. In your 10-minute talk, you should aim to have your introduction on a single slide. So before we talk about the slide, look at your watch. See how long it takes to work through this introduction. Are you ready? Let's begin. So today we're going to talk about our research work, which focuses on how amyloid beta oligomers and dimers isolated from deceased human subjects can impair synapse structure and function. And this research is important because, as we all know, AD is a critical health problem. And the disease is distinguished from other dementias by the accumulation of amyloid beta in the brains of sufferers of AD. Now here's what we know and what we don't know. Experiments have shown that neuronal alterations are induced in mice by using synthetic amyloid beta peptides and amyloid beta species from cultured cells and amyloid beta assembly forms. But no one has taken soluble A-beta extracted from humans with AD and used that. And thus the pathogenic activity from the human species of AB is unknown. So in our experiment, we decided to use soluble A-beta oligomers isolated from the cerebral cortex of deceased human subjects that had AD in order to induce the Alzheimer phenotypes in normal adult rodents. And our hypothesis is as follows. We think that soluble A-beta from human Alzheimer's disease brains would impair synaptic function and structure. And we believe that it would do so specifically by the actions of the A-beta oligomers and dimers. Now, how long did that take, that introductory slide? Less than 2 minutes. Less than 2 minutes to set up the entire research problem on one slide. Now you can see how the rest of the talk is set up, and everyone knows what it's going to be about and what you're going to try to prove. That's why this introductory pattern is so powerful when giving a talk. The CCQH pattern is a powerful, effective way to structure an introduction. Any introduction, for a talk or a poster or a paper. Using this pattern, the CCQH pattern, will help your audience understand exactly the nature of your research work and make for a compelling and easy to follow talk.